Hi, welcome back to Discovering Patterns in the Microbiome. In this slide, we'll be talking about visualizing diversity and specifically discussing the differences between principal components analysis, which is PCA, principal coordinates analysis, which is PCOA, and then non-metric multidimensional scaling. So the first thing I want to do is just give you a quick overview of the whole process of going from the input table through beta diversity analysis to the ordination technique. Now, so I want to give a quick overview of the whole process where we start with a data table, let's call it X, and this is where you have samples here, so like sample 1, sample 2, sample 3, and then the columns could be OTUs, they could be genera, they could be any feature, so let's just call them OTUs, or let's say they're genera, so G1, G2, G3, and what we typically do, so we can do lots of statistical tests on the genera, we can calculate alpha diversity, but when we're wanting to measure overall changes in the microbiome structure, we run this through beta diversity. Oops. And this is where we're basically calculating the difference between the overall structure in the community between every pair of samples. So we end up here with a new matrix that is square and symmetric. So it's now sample 1, sample 2, sample 3 in the rows, and sample 1, sample 2, sample 3 in the columns. Then, of course, along the diagonal, what do you have? Zeros. So the distance from sample 1 to sample 1 is 0, sample 2 to sample 2, and so on. And then the rest of these are whatever distance metric we choose. Many of them range between 0 and 1. So let's say, you know, sample 1 to sample 2 might be 0.5. Sample 1 to sample 3 might be 0.75. Sample 2 to sample 3 might be 0.6, and then it's going to be symmetric, as I said. So it'll look like that. Whoops. Now, once we have this big table, there are certain statistical tests we can run on the beta diversity directly. But what we often want to do is to visualize the beta diversity on a piece of paper, so that we can send something off for publication, or so that we can explore the overall variation in the data. So, that's the process that's called ordination. So we'll take that distance matrix and do ordination. And then what do we get? Well, it's going to be a new matrix that is it still has samples in the rows, but the columns are now they're now principal axes of variation. So this would be, say, PC1, PC2, PC3, and we often don't look beyond PC3, although there can be very interesting things in PC4, 5, 6, 7, 8, and so on. So the topic that we're discussing now is how to do ordination. On the beta diversity side, we already mentioned unifrac is great when you want phylogenetic diversity, when you're dealing with OTUs. If you have genera or other features, then we often use Bray-Curtis. And then finally, if you have gradients, we often use the chi-square distance metric. But for ordination, there are several approaches. The most common one in the microbiome is principal coordinates analysis. This comes from the fields of ecology, and it's different from principal components analysis. And so the principal coordinates is PCOA, and then principal components analysis is PCA. Then finally, some people use non-metric multidimensional scaling, NMDS. So we're gonna compare these ordination techniques. Once you've run the ordination technique, then you can plot the samples on PC1 and PC2 and start to see whether, for example, there's some clustering, based on your experimental groups. So what I'd like to do now is discuss differences between PCOA, PCA, and MDS using some code demos. Here we are on the code browser. We're going to go down to Ordination, which we're doing on the Guerrero-Negro data set again. At the top of the page here, you see we're loading the biome package and the vegan package, which we've already done before. And then here are a bunch of commands that we've already used for loading in the OTU table and calculating some distance metrics. I want to point out one difference here, which is that we're changing the OTU table to be relative abundances. So when it's read in raw, it's just the actual observations of OTUs. So you saw this OTU once in this sample, 40 times in that sample, and so on. And that's going to cause problems if samples come from dramatically different sequencing depths. So this command right here with sweep is basically saying, take the OTU table, the one refers to rows instead of columns, sweep across each row, dividing by the sum of that row. And that's going to make each row sum to 1. Now, first thing we're talking about is PCA. Outside the field of ecology, this is a common, the principal components analysis is a common technique for doing dimensionality reduction. It turns out that it's basically equivalent to doing principal coordinates analysis, which we've been using so far, with Euclidean distances. And as we saw in a previous video, Euclidean distances are generally not as good for microbiome data. In fact, sometimes they're downright horrible. So PCA tends not to be a good thing to do with the microbiome. PCA takes the raw data table and goes straight to the ordination. There's no beta diversity in between. But it's equivalent to having Euclidean beta diversity and then principal coordinates. Now, I'm going to calculate it here just for demonstration purposes. So the way we do this is PCA doesn't work if you have more features than you have samples. So we're going to pick out just the 10 dominant OTUs, which I've done here. So basically sort the OTUs by their column means, so what's their average abundance. I want it decreasing, and this order function is going to tell me the order of the columns in decreasing order. Then we take just the first 10 of them. And now here's where we do principal components analysis in R. So we take the OTU table, pull out just those 10 dominant OTUs, and run princomp, that's the function for PCA, and the $ scores is going to extract the scores matrix from the result. So we store that in this pca.otus variable. Then for comparison, we're going to calculate Euclidean distance on the same subset of the OTU table, and then do principal coordinates analysis, or CMD scale in R. Then I will plot the first axis of principal coordinates in the principal coordinate analysis of Euclidean distances. That's the x-axis. And then the y-axis, we're going to show the first axis of principal component scores using principal components analysis. And here's what you get. So they're identical up to a scaling factor. So this validates the fact that it's basically doing the same thing. The next technique we're going to look at is non-metric multidimensional scaling. This is NMDS. We can do this using the vegan package. And we're going to run this on the OTU table. And then we have to pull out the points matrix. That's the actual scores on PC1 and PC2 for NMDS. So here's what happens when you run it. And then we're going to use the same approach we used in the beta diversity video for making the plot. So we're making this little set of colors that range from red to blue, and then we're going to color by which layer the sample came from in the microbial mat. Then we're just plotting here the first column of the non-metric multidimensional scaling on the x-axis, the second column on the y-axis, and here's what we get. This is basically the same level of quality for recovering the gradient of depth along the first axis. You get a little bit less of the horseshoe effect, and you saw that very nice arch in the other analyses. Whether that's a good thing, I'm not sure. If it's as likely to recover gradients. I know it's not as likely to recover gradients as the chi-square distance in general. So we can do Pico-A on the Bray-Curtis distances. We've done this before. Just for comparison, here's what we get there. And we see basically the same recovery along the x-axis, but with more of this arch effect, which we're going to go into later. In general, people use Pico-A much more often in microbiome analysis than NMDS. So I recommend using Pico-A. No one's going to complain about it. And the way NMDS is calculated is a little bit hacky. It basically keeps doing this iterative thing, and then it does it a bunch of times until it gets the same answer twice. At least that's how it's implemented in the vegan package. So whereas principal coordinates analysis has a much more reliable statistical foundation. One interesting thing you can do once you have an ordination plot is to ask how the different features, so those are the species, genera, OTUs, etc., vary along each axis. And there are various ways to do this. So one reason why people often use PCA, this is principal components analysis, is because it automatically gives you what are called the loadings of the features along the first axis, second axis, third axis, and so on. So you can easily plot where those features are on each axis. This kind of plot is called a biplot, where you've got one set of points for your samples and another set of points for your features. You can also do this with principal coordinates analysis. It doesn't have quite the same statistical rigor as doing the loadings in PCA, but then again, you get the benefit that you're using whatever distance metric you want. So I'll show you how this is done typically in principal coordinates analysis, or for any ordination technique other than PCA or correspondence analysis, which we'll talk about later. And what you do is you basically treat each feature as a weighted average of all of the positions of the samples that feature is in. So to do that, we're going to renormalize the OTU table by the columns so that the columns sum to 1, which means if an OTU is highly abundant in, say, sample 1 and just a little bit abundant in samples 2, 3, and 4, then most of the position of that feature along each axis is going to be determined by sample 1, by the position of sample 1. Then we can just use a matrix multiplication. So we multiply the transpose of that feature table by the principal coordinate scores, and that sums up and gets a weighted average along each dimension. Then we can plot the principal coordinate scores again. That's the same command we had earlier. And we'll use this points command to add the weighted average positions of the OTUs in each axis. So here we have weighted average. This is the matrix that came out of that matrix multiplication. So weighted average is taking just the top 10 OTUs. Remember this OTU.ix variable we set earlier to have just the top 10 OTUs. So we're going to plot the first axis scores by weighted averages, the second axis scores. I'm going to make the points a little bit smaller than the sample scores, and I'm going to use hollow black circles. That's PCH1. Then I'm also plotting at the same positions the names of those OTUs using the text command, and here's what we get. So these are the samples in color, and then the OTUs that are most dominant as weighted averages of those samples. And then the text that's there is just the OTU identifier. So that these are closed reference OTUs, so those are the green genes identifiers for the OTUs. You can also do principal coordinates analysis in QIIME, including 3D coordinates, which is really useful, some other nice aspects, and then finally you can also do the biplots in QIIME. So now we're going to switch over to the command line. This is no longer in R. This is on the Unix or Mac command line. And the way you have to set this up is to run first beta diversity. And we've done this before. By default, this calculates the weighted and unweighted unifrac beta diversity matrices, but you could also run, if you do beta diversity dash S will show you all of the metrics that are available. So you could run chi-square, you can run Euclidean, you can run Bray-Curtis, and so on. So we run beta diversity. That gives us a distance matrix. We do ordination by running principal coordinates, so that's this command. And there we just input here we're using unweighted unifrac, and then we output the principal coordinates analysis file, which I've called picoa underscore unweighted blah blah blah. And then the way that you make the 3D plots is with make emperor. So emperor is the name of the package that's used to visualize the 3D plots. So you use the QIIME command make emperor, and this is one of the most popular commands in QIIME. You input the principal coordinates analysis table. You pass it your metadata file, the mapping file, so that it can color the points by different metadata features. You give it an output folder, which I called 3D. And then here I've set number of segments. So this basically controls the smoothness of the spheres of the points. And you can go up to 14. If you have a lot of points, like a really large data set, then I recommend not changing this value. But otherwise, it's nice to make them a little bit smoother. So you get this output table when we run this command. This is going to give us an output folder, and then we can open, there's an index.html file that we can open in our favorite web browser. So here we have this in, I have this open in Safari, which is not my favorite web browser, but it opens more easily. And then you can actually rotate the points around in 3D. And here we see the gradient. So let's color these, click on the colors tab, color by the end depth of the sample. And here you see that that they're ordered by depth and they're kind of winding around in 3D space like this. We can also turn on sample labels if you want to find out which sample is which. And you can also change which axes you're looking at. So if I wanted to see, so PC1 is the dominant gradient. If I want to compare that to PC4 and 5 and see if anything interesting is going on, you can do that too. And in large data sets, you sometimes will see very interesting things. Parallel axes is another very nice way to see if there's any interesting signal in the later axes of variation. For example, we might look at a data set that had three treatment groups. You might see that PC 1, 2, and 3 are explaining the difference between, say, one treatment group and the other two, plus some other variables like, batch effects in your sequencing run or whatever. But if you look at the parallel axis plots, you might see that somewhere out in PC4 or 5 or 6, all of a sudden the other two experimental groups get separated along that axis. So that would tell you you might want to go in and visualize those axes. So this is just a nice way of exploring your data, and then you can take screen captures and use these as figures in a paper. As I mentioned, you can also make biplots in QIIME. You need to have a tab delimited taxon summary file. So when you run summarize taxa, this QIIME command, you get out a biome version and a txt version. Then here I'm asking it just to produce level 6 taxonomy, that's genus level. We run make emperor. This is the exact same command, but here we have -t OTU table L6. We open the resulting HTML file, and what we get is a similar plot that shows not just the samples, but also the top 10 or so taxa and where they are. And these are genus level taxa. If we go to the labels tab up here, we can turn on the biplot label visibility and get labels put on those. It's a little unwieldy because these labels are really long, so it's hard to tell what's what. But if you wanted, you could go in, you could open the genus table in Excel or a spreadsheet app and easily just edit the names of the genera to be shorter for a nicer display in that biplot. So that's basically a summary of what you can do in QIIME with various distance metrics and principal coordinates analysis, including generating biplots.